Good morning. It's November 6th, and this is your daily brief in science. Here's everything you need to know. Researchers at UC Santa Barbara have made a significant discovery regarding the mating habits of male mosquitoes. Their study reveals that deaf male mosquitoes show no interest in mating, highlighting the essential role that sound plays in their courtship behavior. Published in the Proceedings of the National Academy of Sciences, the findings indicate that genetic modifications that eliminate hearing in male mosquitoes completely disrupt their mating behavior. Emma Duge, a co-author of the study, emphasized the unexpected impact of losing just one sensory modality on mating, which is typically influenced by multiple sensory cues. In controlled experiments, while hearing male mosquitoes successfully mated with females, the genetically modified deaf males exhibited no mating behavior even after 3 days in the same enclosure. This research could significantly enhance the sterile insect technique, or SIT, which involves releasing sterile males to mate with females, ultimately preventing reproduction and potentially controlling mosquito populations. By targeting the TRPV-A gene, researchers believe they could improve the competitiveness of these sterile males, making SIT a more effective strategy for population control. Understanding the role of sound in mosquito mating could lead to better methods for suppressing populations and preventing the spread of diseases. Mosquitoes, particularly the Aedes aegypti species, are major vectors for diseases like dengue and Zika, affecting approximately 400 million people globally each year. Since only female mosquitoes transmit these diseases, keeping them unfertilized could reduce both the population and the risk of disease transmission. While mosquitoes are often viewed negatively due to their role in disease transmission, it's important to note their ecological significance, including serving as food for various animals and contributing to pollination. Previous genetic modification efforts in Brazil have shown promise, with teams successfully altering mosquito survival and mating capabilities to mitigate dengue risk. Montel's lab continues to explore innovative methods to enhance SYT, aiming to suppress mosquito populations below critical thresholds for effective disease prevention. Bright light therapy, or BLT, is emerging as a promising alternative for treating depression, offering a drug-free option for those who struggle with traditional medications. Major depressive disorder impacts about 5% of adults globally, and while antidepressants are commonly used, they may not work for everyone and can lead to undesirable side effects. This makes the potential of BLT particularly significant, especially given the harmful effects and costs associated with conventional treatments. The American Psychiatric Association has recognized BLT as a valid treatment option for major depressive disorder since 2005. A recent study published in JAMA Psychiatry analyzed data from 11 trials with 858 patients, finding that those receiving BLT had a remission rate of 40%, compared to just 23% in the control group. Participants also showed a notable reduction in depression scores, as measured by the Hamilton Depression Rating Scale, reinforcing the therapy's effectiveness. In addition to being effective, BLT is also cost-effective, generally incurring lower expenses than the variable costs associated with outpatient antidepressant treatments. However, it is crucial for patients to select the appropriate lightbox, as some marketed as 10,000 lux may not provide the necessary intensity if misused. Experts caution against at-home light therapy without professional guidance, stressing the importance of correct exposure, duration, and light intensity for safety and effectiveness. BLT has a well-established history of helping regulate sleep-wake cycles, particularly for individuals with sleep disorders like delayed sleep phase syndrome. Research indicates that exposure to bright light positively influences mood and cognitive functioning by activating retinal ganglion cells, which are essential for mood regulation. A 2023 report from the CDC revealed that nearly 20% of adults in the U.S. have been diagnosed with depression, underscoring the urgent need for effective treatment options like BLT. A patient participating in a clinical trial for a CRISPR-based treatment for sickle cell disease has died, raising serious safety concerns about gene editing therapies. This incident involves Beam Therapeutics' Beam 101 treatment, which aims to utilize base editing to enhance the production of durable red blood cells. The cause of death was identified as respiratory failure, likely connected to the chemotherapy regimen involving busulfan that was required prior to receiving the Beam 101 treatment. The patient passed away 4 months after undergoing the therapy, underscoring the potential risks associated with the necessary pre-treatment regimen. Although the trial had previously reported promising early results, including significant increases in fetal hemoglobin and reductions in sickled hemoglobin, this death may overshadow those findings. As a result, Beam Therapeutics is now facing increased scrutiny as the trial moves forward, which could have broader implications for future gene editing therapies. Beam's CEO has acknowledged the risks associated with myeloablative transplants and chemotherapy, but emphasized the need to balance these risks against the severe health threats posed by sickle cell disease. The patient was among 6 individuals treated in this groundbreaking trial, which focuses on a new form of CRISPR technology. Currently, Beam has enrolled 35 patients in the expansion cohort of its Phase 1/2 study testing Beam 101, with plans to present further data at the upcoming American Society of Hematology's annual meeting in December. This incident highlights the ongoing risks associated with older chemotherapy drugs like busulfan, which continue to be used alongside newer gene therapies. It also emphasizes the inherent dangers of one-time genetic therapies for blood disorders that still depend on toxic chemotherapy agents developed in the mid-20th century. A recent study involving nearly 30,000 individuals has examined the long-term effects of additional years of education on brain structure. This research was based on a natural experiment stemming from a 1972 UK law that raised the minimum school leaving age from 15 to 16, allowing researchers to compare the brain structures of those affected by the law with those who were not. Utilizing the world's largest collection of brain imaging data, the study analyzed MRI scans taken 46 years after the schooling change. Published as a pre-registered natural experiment in the journal eLife on November 5, 2024, the findings reveal that, despite the known cognitive and health benefits associated with education, there was no significant impact on brain structure from an extra year of schooling. These results challenge existing theories that position education as a significant protective factor against brain aging, indicating a need for further investigation into this relationship. Professor Roger Kievit suggested that any potential effects of education on brain structure might be microscopic and undetectable with current imaging techniques. Researchers speculated that while education might initially alter brain size, these changes may not persist long-term, similar to temporary gains from physical training. They further proposed that any structural changes induced by education could be too small to detect with existing MRI technology. Ultimately, the study suggests that while education may lead to temporary increases in brain size, these changes are not permanent and do not appear to be reflected in structural MRI measurements over time. Ring Therapeutics has forged strategic partnerships with ASTAR and the Singapore Eye Research Institute, or SERI, to boost research and development in gene therapy, specifically targeting ophthalmology and oncology. This announcement was made by Ring Therapeutics CEO Tu-Yen Ong during an interview with BioWorld. A memorandum of understanding was signed on November 4, 2024, in Cambridge, Massachusetts, formalizing these significant collaborations. This initiative highlights the increasing interest in innovative biopharmaceutical solutions within Singapore's dynamic research environment. ASTAR, Singapore's leading public sector research and development agency, aims to connect academia and industry, fostering economic growth and societal benefits. Meanwhile, SER1, established in 1997, stands as Singapore's premier institute for ophthalmic research, known for its substantial contributions to understanding and treating eye diseases. Professor Tan Zi Wei from ASTAR expressed optimism about the collaboration's potential to translate scientific discoveries into effective therapeutic solutions for patients. Additionally, Professor Jodhbir Mehta from SERI noted that gene therapy could revolutionize the treatment of previously untreatable ophthalmic diseases, with a focus on developing a novel class of viral vectors. The partnerships aim to create a new class of viral vectors, marking the first significant advancement in this area in over 50 years. Tuyen Ong emphasized that these collaborations will assist in characterizing novel anellovirus genomes and expanding their therapeutic portfolio. The expected outcomes include accelerated development of novel therapeutic candidates and improved bioprocessing technologies essential for their production, facilitating the clinical translation of their anellovector therapeutics. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.